Okay.
Another
Can you-
1 minute we will start.
Okay,
sir.
can you hear me?
Yes, sir. Yes, sir. So, Pharmacological Society West Bengal Chapter and our Technical Associate Inovocare E-Academy, I welcome you all in this academic event. But before going to the subject, it is very important to remember one of the greatest son of this Mother India, Sri Dayananda Saraswati. One very important quote he had made, just remembering his quote, that a value is valuable when the value of value is valuable to oneself, and no religion can demolish that by indoctrination. Nothing can touch it because it is empathy. So it is very important to give stress on this empathy issue. Even we are talking about preclinical drug development, because which is preclinical today is going to be clinical tomorrow. Apart from considering the different animal ethical issues, it is also important to consider this group of drugs or this group of substance is going to be taken by human tomorrow. So a very important topic today we are going to discuss, and we are really privileged to have two very important and very noted teachers of pharmacology with us today. We have Professor Swati Bhattacharyya, Madam, Professor of Pharmacology, now at Raiganj Medical College. I welcome you, Madam. And also we have Professor Naser, Sir, who is a noted clinical pharmacologist and professor of pharmacology, now at Calcutta National Medical College and Hospital. I welcome you, sir, on behalf of this organization. So please start today's proceeding.
Okay. Thank you, doctor. And now I will start this topic. So I also like.
Madam, sorry to interrupt. Madam, sorry to interrupt. I request our respected chairpersons to start the session. Then we can start your presentation.
Okay.
Okay. Good morning.
Good.
Good morning, everybody. Dr. Baisakhi, can you hear?
Yes, sir. Yes, sir. Good morning, sir.
Professor Swati Bhattacharyya is there?
Yes, I'm there.
Yes.
Carry
Okay,
on.
okay.
Carry on,
Okay.
Dr. Naser.
Okay. So today, may I introduce, or Dr. Bhattacharyya, will you introduce?
You introduce.
Okay.
You start, Dr. Naser. You start.
Okay. So we are going to discuss on the particular topic of preclinical drug development, as been described already by Dr. Shambo S Samajdar. So we are going to listen from a very renowned scientist, and who is Dr. Baisakhi Moharana, who belongs to CSIR CDRI. So we are eager to listen from her. All of us, we know that preclinical drug development is a very important topic, and definitely it has got the issues of ethics, which Dr. Samrat has already mentioned. The empathy is very important, and that's why so many regulations of clinical animal handling and animal keeping in use of preclinical drug development. So we think we'll have a very good discussion on this particular topic, which is very important for our post-graduation students for MD pharmacology. So I think they will be benefited from this lecture. So I'd request Dr. Swati Bhattacharyya if she can say something.
First of all, a very good morning. And first, in the first part, I will thank IPS West Bengal for giving me this opportunity to be able to chair this session. As everybody said, it is a very important issue, and already we have this is a fourth session. In the second session, we already had a preclinical study told by Dr. Bhattacharyya from CSIR on this evaluation of potassium and calcium channel modulation in rodent model, scope of medicinal plant molecules for cardiovascular disease. Now, this is the overview on preclinical drug development, and I know it will be a very useful topic for all of us, and for the postgraduate students especially, as Dr. Naser mentioned. So we start our scientific session. Thank you.
Oh,
Thank you, madam. So
Thank you.
with all our chairpersons permission and requesting our speaker to please allow us, because we have our president, sir, Professor Tapan Kumar Mondal, sir, President of Indian Pharmacological Society West Bengal Chapter, with us. So, sir, will you want to share some important thoughts on this topic, and we can start today's proceeding then?
Thank you, Shambo. At the outset, I will welcome today's speaker, Dr. Baisakhi Moharana, to accept our invitation to deliver a lecture on this topic, very important topic, that is preclinical drug development, which is highly required for the PG student, particularly MD pharmacology, MBSc veterinary pharmacology, and also M.Pharm student. So on that basis, we have selected this topic, and I think it will be helpful to all our PG students. I will welcome today's chairpersons, Dr. Swati Bhattacharyya and Dr. S.M. Naser, to chair today's session. I also welcome, I am seeing that there are some old and retired persons, we can say, so pharmacologists. So Professor Rabindranath Chattopadhyay, Professor B.K. Rai from Ranchi Veterinary College, Professor K.K. Sardar from Odisha Veterinary College, and so on. And all the students of postgraduate level from veterinary science, medical science, I welcome you all, and I think you will be benefited from this lecture. Thank you, Shambo. Now you can start.
okay, can I now start?
Yes,
Okay,
yes.
okay, thank you so much, sir, and for this opportunity. And really, with so many eminent persons, I feel like I'm new to this field, and not new exactly, but I will tell like my experience and whatever I knew in this topic. And so I'll start this topic, that is preclinical drug development. So when I actually belong to this CSIR CDRI, that is Central Drug Research Institute. This is the drug research. I think this is the first drug research institute by Pandit Jawaharlal Nehru, and this institute has been more than now 60 to 70 years. So it's actively involved in drug development and preclinical studies. So been here for 4 years. So here I learned how these studies and how the things are going on here, from drug design to up to preclinical testing, then it will go to clinical trial. So I'll point out some overview, as this is a academic class for PG students. As a young professionals, PG students, whenever they are doing some research, they are aiming at seeing some target, or what are the inflammatory changes or something. This is the preliminary things generally we see. So after that, we see for which will be the druggable target. Many targets will be there, but what are the druggable targets? How we can aim it, and how we can develop one drug. This is not a easy process as it seems. So it's taking nearly, here it takes around once career, sometimes it's 18 years, 20 years, or 15 years, like that. So depending upon the drug, and also it used to fail at the end. So this is a very important topic, preclinical drug development. I will, for PG students, it will be overview, like how the things are being done, and I'll tell my experience here. So like introduction, so preclinical testing or assessment is the interlink between drug discovery and clinical testing and the ultimate availability of the drug product to the patient. So in clinical, all these things first start with drug discovery, then drug development. Then after the drug development, there will be preclinical testing. After preclinical testing, then all the clinical trials. Clinical trials, I will just tell in the brief. As I discussed, it takes around 12 to 15 years of development cycle to go from lab to clinics. So many compounds in the first phase there are many screening procedure where we screen the different libraries or different molecules, also phytoextracts. And from that, with thousands of screening, only 1 to 2 compound maximum can go to phase 4. Okay, this is a very time consuming as well as so many money also being spent here, like so many budget we have to apply and all other thing. So it's a very costly affair as well as very complicated, complex, as well as it's a very risky task also when we go. So this development is the entire process of taking a newly discovered compound or drug through regulatory approval to the point of marketing. During the development, the new drug or the compound should adhere to the high standards in the conduct, analysis, and interpretation of preclinical and clinical studies for its smooth passage through the regulatory approval phase and eventually to marketing. So different phase: discovery, preclinical, testing. Okay. So here, so first phase is drug target identification. In drug target, so that is called druggable target. Many targets will be there, like that's what I say. Maybe it's a G protein, maybe it's a like any ion channel or anything. So this one first has to like have a kind of like effect. Okay, so we have to see how much it's if we are targeting that particular protein or receptor, so how much that is affecting the downward signaling or how it's affecting the physiology of that particular any disease or condition or something. Then we have to validate the target. Means we'll use some, like for example, maybe some siRNA or something like any antisense technology, and we have to see if we are kind of doing knockdown study or something. So how the particular thing, that protein expression or thing is changing. Okay, so hit to lead identification. Lead identification is very important thing, because so many compounds we have to see the lead compound. Lead compound will be effective in mostly nanomolar range. It's not like some mg or something when in vitro or screening this thing will do. So also it can be an mg thing, but we have to compare with also with standard drugs available. So how and the available in market. Then we have to optimize the lead. Then we will go for preclinical and clinical development. So lead optimization and this thing. So in between, when the particular lead will be developed, okay, so within thousands of molecules will be there. We'll screen everything. For example, a receptor assay, or we'll do some phenotypic assay. So there we'll use thousands of molecules. There we'll get 4 to 5 or sometimes it's 10. We'll get some lead compounds. Okay, then also the active ingredient, that is also called the NCE, new chemical entity. So that one has to be tested like in a GLP facility or in a regulatory facility, so that the data whichever we got, that should be like that can be approved worldwide. So that is the main condition, like one should have one GLP facility to do the preclinical testing of that molecule. So then only the data will be like accepted throughout the world, and the particular thing and the clarity, and we can be sure that the thing what we are testing is like in the like future also we can make sure of the data that it's okay, it's correct only, because sometimes data manipulation and other things. So these are very like I can say in drug development is very dangerous thing, because it may pass the first phase or second phase, in third phase or fourth phase, again it has to return. So why to go this far? So we have to be very sure of, like in the first phase, in preclinical itself, we have to be very sure of its activity. So that only we'll go for phase 1 or clinical trial. Anyway, that generally we used to like submit this preclinical whatever data, like we have to file IND, investigational new drug, to FDA. So within 30 days, I think it has to give the answer. Then the drug will go to phase 1 study. So I will tell very brief about clinical trials, because I'm not going to discuss more about these clinical trials. As we know, in phase 1, mostly we give this the lead compound, whatever after preclinical testing has been evaluated, it will go to phase 1. Phase 1, it will be tested in healthy individual, just to see PK study and to see PD, and also to see like what is the tolerated dose. Okay, to see the dose effect relationship here. Here phase 1. In phase 2, we'll go for patient. In few patients, we'll do the test and we'll see the effect, okay, of the compound. Then in third phase, which is a like kind double-blind placebo, it will also include placebo study. So here we'll go for hundreds of patients, and it will be also a double-blind study that the patient or doctor, they will not be knowing what they are taking, and it will be a like a very strict protocols being followed in this phase 3. So the drug which will pass the phase 3, then it will go to phase 4. Phase 4 is post-marketing surveillance. That is, the surveillance means if it is, that is marketing phase. That is the also main phase where many drugs generally they're being withdrawn because of some side effects or some because in clinical trials and all and preclinical study, for example, there will be some headache or allergy reaction. Generally, we never used to we used to forget that to document, like if there is any immune reactions, okay, in animals. So when we do this post-market this phase 4 study, there itself we can like document if there is any like allergic reaction or any immune reaction or any severe reaction. Sometimes some like for example for cancer drugs and all, they used to like have if this kind of like few like side effects, then it is okay, like but something which is related to major kind of like systemic like it affects some systemic our organs, or like to heart, or there is some kidney failure or bone marrow toxicity. Sometimes a severe toxic things, they are generally withdrawn from the market. So these are 4 phases of clinical trials. So here drug discovery, and in between will be preclinical testing. So in CDRI, I also involved in preclinical testing of drugs, mostly in safety pharmacology of the lead compounds. So in GLP facility, we used to conduct the test, safety pharmacology. So I'll deal in the later slide. So finding NC, new chemical entity. First, we have to identify the disease. Okay, what we want to identify. So maybe we will isolate the protein involved in disease. Sometimes we can isolate the protein, or sometimes the structure is already present. We can do structure activity relationship, that is already known structure. So if we know, then also we can predict like whatever the effect. Mostly it is done by this chemistry people, and then it comes preclinical testing in animal models. So in CDRI, we have different animal models, like from in cardiovascular field, in our airway inflammatory field, in diabetes, cancer, in osteology. There are so many, and also like so many departments we are having. So preclinical models, so many available in here. So we used to test in those animal models as well as also in vitro models. So then the major thing is scale up. Sometimes what happens, like sometimes by like serendipity, you can say, like sometimes one molecule is being developed. Okay, so it has some effect, but when we need that molecule in mg or some kgs to do in vivo like testing, then that becomes so difficult. So like the production of that particular lead compound is very important. Like it should be the steps of the preparation, it should be properly like designed, or I can say like the manufacturing process of that particular compound, the purity. That's the major thing, the purity of the compound. It should not contain more than 0.1% of impure, like unknown impurity. It should be 99% pure. To go for GLP, all testing of that particular compound, it should be pure and it should be properly, we can prepare that particular compound, the batch, all the characterization of the particular batch of compounds should be same. It's not like the characters, like maybe the color is changing from every batch to batch. So all the batch should be same before using in animals. So scale up and formulation are very important procedure. Here in GLP facility, we have the storage of that particular compound, also characterization. Here always like pharmacokinetics and those people, they characterize each batch so that we can get the data like the whatever batch we are testing and we are reporting also, that is from the like same compound. So this scale up formulation, then we'll go for IND, because after filing IND within 30 days, they will give the reply. So it will go for phase 1 clinical trial. So when in human, so it will be tested. So before that, we have to like do the scale up things. So human clinical trials will be done. Then at the end, file NDA. Then we'll go for drug authority approval. So nowadays, failing fast and cheap is an indispensable basic rule of thumb. That is, failing fast and cheap means at the first stage in preclinical testing, if we are failing, that is actually good. Then going and showing some data, nowadays they have seen that their sample size is less and other things. They are a very important criteria nowadays. Like they are making the data to show that like it's acting very good in vitro purpose and in vivo. But when it go to phase 1, in phase 1 itself, it fails. Phase 1 or phase 2, it fails. So failing fast and cheap is a actually good thing. Then going for a like phase 3, which is time consuming as well as it takes so much budget. So this is the thumb rule now. So we have to be very careful. So only we are doing in GLP facility so that everything is being documented, all the raw data. That's the very important thing. All the raw data we are maintaining in a cloud, like in a system, so that there should not be any mistake in statistical calculation or anything. So we have like raw data as well as all the recordings of the software, like whatever we are like using the model or like software we are using to record any cardiovascular activity or respiratory activity or anything or neurological activity. Everything is being recorded and being stored. It's like very password protected, like in GLP facility. That will be useful whenever, like any USA FDA, because CDRI also filing in US FDA the drug approval. So there it has to show the data. So data should be very like clean data. There should not be any mistake or like any like space of any irregularities. That then only it will be approved by like FDA, and also will be also happy that maybe the drug can pass the phase clinical trial phases. So drug discovery is not about only a biologist. So in our CDRI, we have medicinal process chemistry division. So chemist people are there. Also molecular structure biologists are there. In bioinformatics are there. So in silico designing drug, matlab how in silico also we can know like when the drug, the receptor binding study, like docking study, okay, how much energy and docking score and all. So from there also we can predict the action. So bioinformatics people will tell, then the chemist people will prepare the drug, okay, the medicinal chemistry people. So in our pharmacology department, we will do the pharmacology part, like to see the phenotypic effect on that. Then toxicology department will do the toxicity study, as well as they will also see the GLP facility, like how to do all acute toxicity, chronic toxicity, repeat dose toxicity study, as well as pathological testing. Also pharmaceutics people, like do all the PK study. PK study is very important. Like when we are going for human study, the clearance, biodistribution, how much plasma protein binding, those are very important points when we'll do for, because sometimes clearance and other thing like those are very important points whenever we are representing half-life. So PK study and how that's the again thing, blood-brain barrier, and also how it is inhibiting or site enzymes. Those metabolism study by PK people, those are very important studies that that used to be done in pharmacokinetics department. So other than that, so many like other studies like for example, this histopathology and other things. Those are some skilled person are there. So all are done in like in like our GLP facility, all toxicity study. So what is drug? So when we are discussing about drug, so drug is a substance or product that is used or intended to be used to modify or explore physiological or pathological states for the benefits of the recipient. Okay, so it may be for diagnosis or maybe some for treatment, or they should be selective. They should act specificity and selectivity. Those are very important points. Okay, it should act like whichever we are aiming at the receptor, it should act on that particular receptor and should have less effect on other receptors. Generally, they used to act, but we have to also see secondary pharmacodynamic study, those are called. So we have to see like if it is having some other effect other than our like beneficial effect. So 1 minute. So why the drug regulation and other thing were important? So because in FDA and other thing, they came into play when first time in 1938 with an incident that at that time during 90s that producing antibiotic was a very big challenge. So in a large scale. So one company that is from I think Tennessee or some company, they prepared this elixir sulfanilamide. They used the solvent diethylene glycol, and they have like a raspberry something that this thing. So people used to take it rampantly in the sense whoever they need, they also it was known diethylene glycol may cause kidney damage, although but they used that chemist used this solvent. So many people, more than 100 people, they die because of that, because at that time there was no regulation. So whatever the solvent or this thing not been used in animal models. So at that time, President Franklin D. Roosevelt, they have signed this FDA Cosmetic Food and Drug and Cosmetic Act, where it has given power to FDA to regulate all the drugs, like whatever like coming, they regulate very meticulously all the toxicity study, all the pharmacodynamic study, PK study, then they will approve. So there is another tragedy that happened, that is thalidomide, this thing, tragedy, when it was called the wonder drug because it was used as antiemetic. So at that time also it was not known drug may also pass placental barrier. So the test there at that time it was tested in animal models, but it was not known that it can also pass through placental barrier. So at that time also, so for the European Union and European countries, they also made their rule that drug, whatever the drug being coming to the market, it has to be passed through some regulatory protocol so that there should not be any like adverse effect. So here, what are the main pillars here? So drug discovery, preclinical development, clinical studies, and marketing, post-marketing activities. These are main pillar of drug development. So how drug works. Drug acts on the receptor. So I'm not going to details on this thing. So why we need for drug discovery? Because now there's new diseases are coming. Some are low efficacy, some having some side effects or the cost of therapy, also to have a industrial activity. So we need for new drug discovery. Then what are the targets? Targets mostly now most of the drug they are being coupled, this G protein coupled receptors. Some are enzymes or hormones or ion channel or some are for nuclear receptors. Target identification is being done through like genomics, proteomics, or bioinformatics data. Also, we can identify the target like in our basic research when you do, we can see like some like how the particular thing is being changed if we can knock down something. So there also we can do some like analysis and we can see like we can identify like maybe this target can act and we can proceed. Then target prioritization or validation. Then this validation generally being done like we can use some like different molecular assay as well as also we can so some antisense technology there also we can validate the target. If like confirm the changes, like whatever desired changes we are like expecting, so we can see the change there. So lead compound is a very this term which is being used in our CDRI very because this is the compound which will be given a code name to go for next stage of stages like preclinical testing and other thing. So lead compound is a compound from a series of related compounds, because many compounds with different side chain or different compounds, same compounds, thousands of compounds, from that one lead compound will be which will be having some desired activity, and that also having at a nanomolar those things we expect should have an effect on nanomolar or molar like millimolar range, and modified to produce another molecule with a better profile of wanted properties to unwanted side effects. So it is generally chemists like how to stereochemist. Sometimes they produce C strands, and sometimes the organic chemists they use some now this chlorophyll, some green synthesis and other thing. So many this is the phase where chemist people generally involve and they give us the the molecule. So identification of lead compound, maybe some medicinal plant to isolate some active compounds from the medicinal plant, or from many synthetic compound libraries are nowadays like there is buying, and CDRI is also having some libraries, and some repurposing of drugs like many cancer drugs are also repurposed for different purpose, and some modulator or something, and fragment computer aided designing also being done. So properties of a good lead. So good lead has many properties, but like for example, it should not interfere with P-cype enzymes or P-glycoprotein, which is a flux transporter. It should it should be soluble in water, and stability, other thing, they depend upon the disease condition, like or the mode of like oral, we are giving oral or through IV accordingly. So it should have selectivity and specificity. Lead optimization. So we can alter the structure of the lead candidates to improve properties. Sometimes cis is not acting, trans is acting, or else we can add some hydrogen bond, some methyl groups, and early chemist people used to add different groups, and we used to do the analogs and we used to test it in our biological system. There are many high throughput screening systems are there, like some beta arrestin assay kind of thing, and so many like inflammatory protein ELISA kits are there, and also ex vivo this systems are there. Also we use in our phenotypic study in our model, disease model. So then structure activity relationship. So because structure activity relationship means we know the like different structure, they have some activity. So depending upon the structure of the compound, we can predict the activity. These things. So preclinical research. So after the lead compound, then we'll come to preclinical research. Generally, preclinical research consists of first thing. First, whatever the lead compound will come, it has to be tested for its pharmacodynamic, means its activity first. Then after getting the activity, it will be tested for its safety. So there it comes toxicity study and safety pharmacology. Those are vital points of preclinical research, and PK study. PK, PD, and safety pharmacology and toxicity studies. So most toxicity study and safety pharmacology, FDA requires the GLP condition. So our CDRI is having this good laboratory practice, this GLP condition, so that the data whichever will be will be obtaining, it will be accepted worldwide. That is the main aim of GLP. Everything is being documented, everything is being available, like for future for so many years. So there will be raw data will be as such. It will be there. So there will be no like the tampering of the data there. That's the main of preclinical research. Then so different animals, when we do animals testing, generally we go for two rodents and non-rodents. In non-rodents, we go for like sometimes we go for like this dogs or monkeys. We have very good primate facility. I think ours is a very active primate facility in our country. Our CDRA is having monkey facility. So we do toxicity studies in primates to go after, like when the drug has to go for IND. Before that, we have to do testing in primates. So preclinical research. So that's what I discussed. Most important is PKPD, toxicity study, and safety pharmacology study. Those are the preclinical main points, those things. So time required is like so much, it's depending upon the particular, like the molecule and also the model, it depends upon that. Types of studies includes in preclinical trial. Screening test and isolated organs or bacterial culture. So many here also a toxicity study. For example, in vitro we do EMS test, we do COMET assay, we this mutagenicity, also reproductive toxicity. There are many assay, in vitro assay as well as in vivo assay also we do, and this pharmacokinetics toxicity assay. Then this toxicity study generally go for acute toxicity and the repeated dose toxicity study. There itself either single dose is being given, another repeated dose, dose is being repeated. Maybe it's for 28 days or sometimes 3 months or 6 months, depending upon chronic or this thing. Carcinogenicity studies generally go for 24 months, and reproductive toxicity generally done in rats. So this toxicity study generally gives the no observed adverse effect level. So which will act as a safety margin. That's the main point. And acute toxicity, that's what I told. There itself we can see the pathological changes after like only once dosing. Then we'll see what are the effects on the normal behavior of the animals, as well as after we'll do the pathology. We'll see if there is any particular, any organ specific, any changes are there. So, okay, so toxicity study is being done in our GLP facility. So I have been involved in GLP, many compounds used to do the GLP study. So there we'll take minimum 3 doses, that is low dose group, mid dose group, and high dose group. So accordingly, also logarithmically, we used to take 2.5, 5 times and 10 times of the particular, whatever the LD50 they have given or whatever that they have given. So accordingly, we'll calculate and we used to prepare our study protocol. So preclinical GLP, all preclinical studies are carried out under standardized procedure under good laboratory practice. So this GLP will take another class because it's so many terminologies there in GLP facility and all the principles. So this GLP try to ensure the uniformity, consistency, reliability, reproducibility. That's the major thing. Reliability and reproducibility means whenever it will be done in other site, it can be reproducible. Quality and integrity of chemical. Okay, so the GLP facility generally involved with this non-clinical test. And there in GLP facility, there are study director, test facility management, quality assurance team. So everyone used to work together to have a like to get to do this toxicology test or septic pharmacology test. So importance of preclinical trial to determine the dose, toxic dose, or pharmacological action. It is the requirement of regulatory body for performing clinical trials because FDA needs all this data for giving, like when we submit the IND. So kinetic profile of the drug also, so a route of administration for human for clinical trials. Because for many immunosuppressants, actually when we use limitation of routine preclinical study, because here what we use, in a very like pathogen-free environment. So, but when the drug is being given to a human condition, they are being diseased one, they have some like their gut microbiota and other things. So many things come into play. So these are some of the limitations. Also, the thing is when we go for this immunosuppressants, another thing. So control laboratory condition is very different from human patient in variety of environmental situation. Also in cancer patient or something, generally they occur in old age animals. But when we do it's young and healthy animals, we used to test. So these are some limitations of this preclinical studies. So these are some glossary. I think this is very simple things. I will not go in detail. So pharmacology study means primary pharmacodynamic study and secondary pharmacodynamic study. Primary means we'll go for directly whatever we are targeting the receptor, whether we are getting the effect that particular lead compound when we are giving, we get desired therapeutic like target we are getting. Secondary PD study generally focused on the mode of action effects of relevant compound which are not related to its desired therapeutic target. For example, it is acting on different target and it is causing some physiological activity which is being not expected here. So secondary pharmacodynamic study, these are side by side is being done. And PK study, observation, distribution, metabolism, excretion, toxicity. Here main data are half-life, clearance, biodistribution, plasma protein binding, and the AUC. Okay, then these are the important parameters being decided in PK study. Also permeability, also acting on like CYP enzymes, it is having some effect or not. Then comes safety pharmacology, which is being done in our pharmacology department under GLP facility. So safety pharmacology of different molecules, that is new chemical entities being done. That is definition is those studies that investigate the potential undesirable pharmacodynamic effects of new chemical entities on physiological function in relation to exposure in therapeutic range or as above for ICH guideline. So ICH S7A generally deals with all core battery tests. Core battery test, why? Because it may affect other organ, but the primary, that is heart, brain, and lung, means respiratory effect, cardiovascular effect, and the neurological effect. These are core battery tests. One drug which is going to the market, maybe it doesn't have any therapeutic effect, but it should not affect our normal physiological function. So safety pharmacology is the very important part of this preclinical test. So, what are the objectives of safety pharmacology? Prevention of serious adverse drug reaction, understanding the concentration response relationship, because safety pharmacology we used at minimum 3 doses. Here we see the effect, and this thing mechanistic understanding and support target identification, other things, but main is to prevention of serious adverse effect. These are the aim. Safety pharmacology study, like different guidelines are there. Our, like for example, CDSCO, Central Drug Standard Control Organization, and the FDA also, like the guidelines are there. So different, our like DCG, they generally see all our data. DCG is generally now one phytotherapy for fatty liver disease. Today only came like been approved for phase 3 trial. So different commission are there. So ICC S7A and S7B safety pharmacology for human pharmaceutical. So these guidelines are being, we can also see in online also it's available, those guidelines. They show like what are the things. So drug discovery, after that non-clinical development. So here pharmacology, safety pharmacology, and toxicology, these 3 are the main part of preclinical study. In pharmacology, as we discussed, in vitro and in vivo studies for efficacy purpose. For safety pharmacology, we have core battery tests, CNS, respiratory, cardiovascular, and ancillary studies are there, but these are the main 3, CNS, CVS, and respiratory. Toxicology, that's what I told, acute, generic, chronic. And safety pharmacology being done in different phases, but main is in preclinical so that it can go to phase 1 trial. So this thing I will just skip. Clinical one. So good laboratory practice, that's what I discussed. test system, test item. Test item is the thing which is being studied. Test system means where it is going to be tested. So these are some terminology. And experimental design, that study protocol, like experimental initiation date and completion date. These are very important parameter in GLP practice. Then study design. Yes, test system is the animal model here. So we have to see all the things, species, strain, gender, age, depending upon the disease condition. Use of in vivo and in vitro study. So different ex vivo or in vitro models are there, like for genetic toxicity or this thing. So we go for in vivo study for septic pharmacology. Sample size and use of controls. Sometimes we use 6 animals, but we have to see the power of the experiment. But generally, for this thing, we use minimum number of animals because this is septic pharmacology study, and also negative positive control we have to add according to the whatever the sponsor or the person who has prepared the particular lead compound. According to the demand, we will use some negative and positive control group. Then route of administration, we can use oral or IV according to the like if it is a suspension, or the route of administration which will be used in human also. So we have to see the expected clinical route of administration. Then dose level or concentration, this thing. So that's what the effective dose into 1, either 2.5 or 5 or 10 times. Generally, we use different dose for like to see all the effect, pharmacodynamic as well as in septic pharmacology. So duration of study, generally septic pharmacology study or single dose administration, will do the single time. That's then septic pharmacology core battery test. Those are CNS, CVS, and respiratory system. In CNS, we see motor activity, behavioral changes, coordination, sensory motor reflex response, body temperature. In CVS, we see blood pressure, heart rate, and electrocardiogram. And respiratory system, we use all the tidal volume, all like pause, enhanced pause. We see like inspiratory time, expiratory time, and all the during respiratory things. So this here also it is being described. What are the CNS parameter, what are the respiratory thing, and what are the cardiovascular parameters we generally do. So cardiac behavioral pharmacology, cardiac output, airway resistance, and these are the supplementary follow-up test. These are not very mandatory, but we can also do after that. Means after applying the IND, also we can do follow-up septic pharmacology test. But core battery test is the important test we have to do. And that's what follow-up. Also, we can do supplemental renal, ANS, autonomic nervous system or renal system, also GI tract, like if it's increasing the acidity. These are supplementary tests. If we will not do also, we can apply for IND. But CNS, CVS, respiratory system, these are core battery tests we have to do. We have to show the data, then only it can pass to next stage. Different tests. So in our listing for septic neuropharmacology, we do body temperature, whole body temperature we used to measure, then Octavarimax for locomotor activity, this hot plate for analgesia, this Rotamax for muscle coordination. And follow-up study like learning, memory, neurochemistry, ligand specific binding. These are follow-up study we can do or we may not do, but these are important study we have to do. Different evaluation of gross behavior generally being done. Protocols are there. In locomotor activity, that's what I told. It's Octavarimax and neuromuscular coordination using Rotarod. And body temperature we have to measure, and sensory motor reflex response. Then telemetric recording. Here we used to implant one sensory device, and after implantation we can measure all the ECG data, all the ventricular pressure and other thing using telemetry. Then whole body Fatismography for respiratory study. Here chambers will be there, so in that animals will be kept, and the animals, all the data, generally frequency, tidal volume, inspiration time, expiratory time, peak inspiratory flow, enhanced pause, minus volume, everything will be like recorded here in the compute software. So all the raw data will be there, and it will be also stored in a CD. So we have Oxco point software and the very latest software to measure all the respiratory study protocol. Then also pulse oximetry for blood oxygen saturation. It's also coming under core battery test. Here we measure the blood oxygen saturation data. Here one technique where the non-invasive monitoring of arterial blood oxygen saturation. And these are supplementary safety pharmacology study like renal, autonomic, gastrointestinal system, organ system. Then these are not very necessary. Like if we can do also, it's good, but it's not very necessary to do. Without that also we can apply. But here as a follow-up study also, we do some basic study here. So conditions under which safety pharmacology studies are not being needed for locally applied agent like dermal or ocular. For biotechnology derived product, they are very specific receptor binding, so we can do the the endpoint of the toxicology also can be considered. So biosimilar things or new salt having similar pharmacokinetics and pharmacodynamic properties, we can do we can also predict the the adverse effect, or accordingly we can also do repurposing, for repurposing drug. Then who are safety pharmacologists? These are very simple things. Then these are some reference. And I can say like different area where CDRA is working, like malaria, leishmania, like parasitic infectious diseases and lifestyle disorder, like diabetes, hypertension, depression, neurodegeneration, cancer, reproductive health, osteoporosis. These are different area where CDRA is being actively involved. And we have like different clinical trial under different with KGMU, SCPGI. We have some collaboration. So with them, we do our phase, like all the phases of clinical trial, mostly phase 1. And these are some under that our animal house is NGCMA approved. So we do all the GLP studies here. Different industry people also approach for their molecule, and we do. Different animal models are there in our like CDRI, mostly good facilities, primate facility. Many industry people also, they come for this primate facility we have. So for toxicity study we use. So different drugs, these are very few drugs I have shown. Many drugs are also in market, like Siles and Chroman, and this skin mine, Bacopulmonary, some extract, and Arti-Ether for anti-malaria. Now also many to not be very complicated, other more deep into the say overview study. So anyone I want to end my slide here, presentation. Hello.
Thank you, madam. Thank you once again.
Okay.
So for this wonderful deliberation. So now may I request our chairperson, sir and madam, and also Professor Tapan Mondal sir is there, and Professor Shantanu Kumar Tripathi sir is also there, to please have some discussion on this topic, and there are some questions also.
Okay.
You can address those questions also.
Okay.
Over to you, madam. Naser sir, are you there? Naser sir?
Yes. Can you hear?
Yes.
Am I audible?
Yeah.
Thank you, Dr. Moharana, for your excellent and elaborate deliberation. And I think it is very useful for the students. Now we can see some of the questions in the chat box. So if we can discuss on those. So Shambo, will you please help to just Shambo, hello.
Yes, sir.
Yes,
So
yes.
I think there is some questions and comments made by Professor Shantanu Kumar Tripathi, sir. So, sir, are you there, sir? Do you want to comment here?
Yes, I am there, but then I can raise those questions and start the discussion. I have put a couple of questions and comments.
Okay.
Yeah.
So,
You
sir
can read
has
them and
asked.
discussion can start.
Yeah, sir has asked that why is term non-clinical development more preferred than preclinical development?
No, the term is non-clinical means the whatever the being clinical means patient bedside. Always the term clinical means patient bedside. So before the clinical stages, we do whatever the test. Those are called preclinical tests. So here we use always preclinical development and other thing. So like I'm not sure non-clinical development. I'm not sure. We use here more preclinical development. So like sir can explain if like sir, because we use preclinical more the terminology because it's
The reason why non-clinical is more preferred because many of these experimentation continues while the clinical development has started. Okay. By this I mean not necessarily all the so-called preclinical development stages or experiments are complete by the time you start clinical development. So there are some so-called preclinical development experimentation that continues in parallel with the early phase clinical development. And that's why these days we prefer non-clinical development, which of course will cover preclinical and also paraclinical developments. Okay, that's one reason.
Okay, thank you, sir.
Right.
Another thing I think, sir, you have mentioned.
In the US FDA, you will find that they do not use the term preclinical any longer. They always say non-clinical.
Okay.
So probably another question was from Professor Tripathi was there any alternative to preclinical drug delivery, preclinical drug development, alternative model other than animal use? Probably sir wanted to mention it. Sir?
Hello.
Yes, let the speaker tell us something about that. That apart from because of we know of the 3R or 4R principles.
Yeah.
So what are the alternative strategies? Some of them, of course, you have touched upon. In silico and in vitro. Use of human cell culture.
Those things actually always this question arises, but as a like whenever we do all this testing, we only know because it will go to human use. If we'll do in vitro and in silico, it's like predictability and how it will act. It will be very like we cannot like predict what will happen. So in vivo definitely we have to go. I mean, the questions can arise, but in vivo we have to go for all the pharmacology.
No, it's not really for predictability. In fact, if you use human cell culture, it is going to be more predictable than the species variation related unpredictability. One of the reasons why people go for human cell culture is to improve predictability. The species difference. So what is found in the animal study, whether it is mouse or rat or monkey, may not be always predictable in human setting. So from that angle, it is said that instead, if you not necessarily all the tests that are done in using animals can be actually reproduced or can be actually done in tissue culture, in human cell culture. But then this is one of the reason that predictability is one of the reason, apart from of course animal use ethics, etc. That people go for the use of tissue culture and also in silico. And besides that, there are also the computer assisted systems. Okay, that has also come in a long way. The deep learning, artificial intelligence, machine learning, they are coming in a big way. Futuristically, these are going to actually optimize the non-clinical development to a great extent.
Yeah.
You have also mentioned correctly about the structure activity relationship that can definitely limit the use of animals. Okay, whereby you can predict that there is no point in doing this particular experiment or that particular experiment. You can minimize the reduce. One of the R of the 3R or 4R is reduction. So reduction, replacement, etc.
Yeah.
So time has come when people are giving more emphasis on these issues, trying to find models other than the animal models, which can provide otherwise reliable data, okay, as well as more predictable data. So, but then truly speaking, we have not totally banished the use of animals yet. Gradually, we are shifting over decades or so. Right?
Yeah, maybe in future because. Okay, thank you, sir. So
there was another issue I just pointed out there regarding diethylene glycol. You mentioned about diethylene glycol.
Yeah.
Organic solvent. So this is organic solvent, no doubt. It has been used since 1937, that sulfanilamide elixir, which caused the tragedy. But then its use is still continuing in many countries, including India. The latest tragedy in the world is the one that happened in India only in 2020, okay, in Jammu and Kashmir. There is a good number of children who were affected. It was actually a cold elixir, common cold elixir for children. And then I don't remember the exact number, some 25 or 35 children were actually affected. And half of them had acute kidney injury. And then a number of them died. So there was an inquiry, and finally it is found the diethylene glycol as a solvent that has been used in that pediatric elixir was much higher than the admissible limit. So that company has been brought to book by CDSCO, and but then children lost their life, and this continues. If you look at the history of use of diethylene glycol since 1937, almost every 5 or 10 years, one or the other country reports this. China, Africa, okay, and different other parts of the world, there are series of such reports, but still we are continuing. Some of the regulators have been very stringent and strict, and it has limited its use. But then in India, we are still using it. Thank you. Over.
Dr. Baisakhi, with the permission of the chairman, just I want to know from you, which animal is suitable for generation of the data during drug discovery comparable with the human data? Rodent or non-rodent, like that?
Sir, actually for that reason, we use two species, rodent and non-rodent. Because like whenever we go for bigger species, we have to like when we'll see all the genetic study and other mice and rats are also they are very close to human species, but there also again we have to go for one primate study or something. So because our GI tract and other thing like we the similarity, so we'll go for one bigger species as per the rule. So
yes, I know that nowadays our planning on rat and mice study it has been deferred to your primate. So primate is more closer to development of the data, which is comparable with the human. Apart from this, our reputed scientist in the field of pharmacology, Professor Santanu Tripathi, he was also in this opinion, there is any alternate method. So alternate method, it is very difficult to say. Every time we are trying, in case of your tissue culture, in case of animal study, but there is no till date suitable alternate for your preclinical study, excepting your animal study.
Yeah, because I am dealing practically, because we are dealing every day practically this thing. So I know what are the issues. In vitro, we are every day in our lab, we are doing the screening with human, this tissue culture, ex vivo culture, cancer also, we are direct from patient genograft, cancer models are being there. But still also we have to go for in vivo study. like in animals. So like this can be the future, like maybe in future we can reduce the animal use, but now like we are very much dependable, I can say, to animals.
Okay, thank you.
Mhm.
Chairman, sir.
Yes. Sir, actually there are Shambo, would you
Anyone is there who can? I think Dr. Chando is there, Dr. Tapos sir, they want to say something.
Yeah, once Antanu sir has written zebrafish. I have also worked in zebrafish model, but still now zebrafish has not been used till now, like for all safety pharmacology. Now it's in like developmental stage, all the all the tests in zebrafish. So another question, I think someone is.
No, actually, I
Yeah.
thought like mentioning it because it is an academic this thing and
Okay.
young graduates are there.
Okay.
So that's
Okay.
why I thought that it is better to remember zebrafish role of.
Yeah. I have worked in zebrafish, so
And that is found to be
yeah. Okay, no, it's
Yeah.
very much its genetic similarity with human.
Okay, there probably there are some questions in the chat box also. So
precision drug discovery? One is precision drug discovery.
Yeah, that is, I think, related to genomics and like the genetic target and what I understand precision means this thing like to see the particular target and
Okay.
genomics and drug discovery mostly what I understand. Precision means direct, but the whatever target we are identifying, it should be a like precise one and like mostly say bioinformatics term or something like okay.
Bioinformatics. Another one is how to know, how do we know if any chemotherapeutic is affecting on immune system or immune status?
Okay.
So to know the immunological derangement.
Yeah, cytokine studies being done for chemo profile and the thing, but it's not coming in safety pharmacology exactly. Immunological profile, when we do pharmacodynamic study, we for chemotherapeutics, we generally follow all the cytokine study, and those are like primary pharmacodynamic study we used to do. Safety pharmacology mostly it's for is it safe, mostly vital points, core battery test generally we do. So in our CDA we do that. So
if you can elaborate a little about the distinction between the toxicological studies and safety pharmacology studies.
Yeah. See, toxicology means toxicity study means one dose will give a single dose or else multiple dose will repeated toxicity study to see the toxic effect of the particular dose. Safety pharmacology means in toxicity we'll see the gross behavior, means gross morphology, and after that histopathology will see organs and other things. And safety pharmacology here we see how safe it is, mostly on physiological functions. Physiological function here important is cardiovascular, CNS, and respiratory. Also there are renal and NS, also GI system is there, but we majorly follow these three studies as safe. This should be safe. It should not cause any effect on physiological function. That is coming under safety pharmacology.
Dr. Baisakhi, just a
Yes.
minute. In case of toxicological study, we are using the dose which is harmful to the body. So that dose is being selected and we are doing toxicological study. But in case of your safety study, what will be the dose? That toxic dose or our therapeutic dose?
Whenever.
Sir, okay, I have a point for the IPS model. Sir, you just finish, then I want to tell.
Yeah. No, sir. Actually, because I am involved in safety pharmacology, the sponsor who has prepared that molecule or has tested the in vivo efficacy, they will give us the efficacious dose. So accordingly, then logarithmically, we'll do at least low, medium, high dose, like in GLP study. So 2.5 times one dose will take 5 times and 10 times. Generally, we'll do that dose. So
that is your effective dose. Effective dose multiplied by 2.5, 5 and 10.
Generally, we used to do like
so
that.
So on that dose basis, we are seeing how far it is safe to the body.
Yeah.
Is it not?
Yeah.
Yeah, sir, with your kind permission, just I want to make one comment for the in vivo models.
Okay.
Dr. Mondal, is it possible to make one comment?
Yes,
Yes,
yes.
yes.
Okay.
Yeah,
Please,
okay.
please proceed. Please proceed.
Yeah, actually, we discussed that human cell lines or this ex vivo studies with human tissues like brain slice, liver slice. A lot of data are there, but one we have to keep in mind, suppose we are doing a study in human cells. Okay, it will be having much better predictability than the data from the animal cells. But when you get a observation from an in vivo experiment in an animal, this is the outcome of the totalitarian effect. Not only the one kind of tissues, the drug is subjected not only for the metabolism or some other agonistic, antagonistic. So there will be different kind of tissues and organs involved, and we get the overall outcome of the response. So cells cannot complement, completely replace an animal studies. Number two, I want to tell, I think many of you are aware, these days, not only very new, but it is going on. This is called humanized model, right?
Yeah, humanized model.
Dr. Durga Prasad Mishra is
DP
scientist.
Mishra.
He's there in CDRI. I
Huh.
have got good contact with him. He has got some nice models, humanized.
Yeah, humanized model.
Yeah, what I'm telling, he has got the mice with the tumor that is actually a human tumor. The tumor
patient xenograft.
Yeah. I am telling the tumor breast tumor tissue has taken from the human, and the tumor is getting developed in the mice.
Yeah.
So those things are working on, but it is still we have to go a long way to completely replace the animal testing. Thank you.
Probably Dr. Taposhar is going to say something. Dr. Taposhar is here. Raise the hand.
Yes, he's there. I think you have to unmute Dr. Shor.
Sir, I have written the question in the chat box. So I request Dr. Shambo to please read the question. Hello. Hello, is it audible?
Yes.
Sir, I have written the question in the chat box. So I am requesting you to, Dr. Shambo, please read the question.
Dr. Moharana, can you see the chat box?
Yeah, I can see.
Will you please respond?
Yeah, a range of therapeutic concentration of a new molecule. See, for example, we always use in like from millimolar to nanomolar range. We will start the concentration dose effect relationship for in vitro study. So we'll not go in mg or something. We'll prepare 1 mg, then we'll prepare the stock solution, and we'll dilute it up to nanomolar range, and we will see all the effects, like different, like G protein coupled receptor assays, or like other like for example like TNF alpha, like inflammatory assays or anything. For in vitro study, we have to do the dose response, because like we'll do minimum dose, then we'll increase the dose, we'll see the like this thing EC50, and we'll see like how the effect we are getting. So for in vitro study, it's not a like we have to, so from minimum to up to 1 mg maximum. So is it the answer? Like anyone can.
Chairperson, I think you may conclude. If there is no question, Dr. Shambo.
Hello.
Yes, sir.
Dr. Naser.
I think we may conclude now, because we had a very good discussion today. So thank you everybody, and I think Dr. Swati Bhattacharyya is here. I'd request her to conclude the meeting.
Thank you, Dr. Naser. So, but it was a very interesting and interactive session. We came to know so many things. And we are grateful to Dr. Baisakhi Moharana. We are grateful to Professor Shantanu Tripathi, Dr. Tapan Mondal, and Dr. In fact, Devabrata Chanda, he also was there, and we could come to know so many things. We are looking forward to attend this kind of sessions more and more. So with that, I thank everybody who attended the session and who all contributed for the session. Thank you so much. Thank you, everybody.
Okay, thank you, ma'am.
Thank you.
Thank you.
Everybody.
Thank you.